Synthetic Biology by Michelle Barringer and Alexis Gomez. What comes to mind when you think of synthetic biology? The term synthetic biology was coined by Barbara Hobam in 1980. It was used to describe scientists' manipulation of living systems through the use of recombinant DNA to genetically engineer bacteria. In other words, synthetic biology was taking a gene from one cell to another using a vector. But in the year 2000, at the American Chemical Society meeting in San Francisco, synthetic biology was described as constructing completely new systems or organisms that are not found in nature. Cells are no longer living things to be manipulated, but little machines to be created. The biological parts necessary to create these new species involve DNA sequencing parts that are combined into devices. These species are then able to perform more complex functions. A recent example of an engineered complex function is the transformation of a sugary seaweed into ethanol, a source of biofuel. The problem with this seaweed is that it contains sugar molecules such as mannitol and alginate, which are difficult for microbes to ferment into ethanol. Scientists at the U.S. Advanced Biofuel Company Bioarchitecture Lab were fortunate to find the bacteria Vibrio splendidus. Vibrio splendidus is a marine bacterium which obtains the genes necessary for alginate lysis in the fermentation of mannitol sugar molecules. Over 20 genes were used from the Vibrio splendidus bacteria and sequenced with the genes of E. coli with the addition of a fermentation pathway and the deletion of some E. coli genes. This new bioengineered E. coli is capable of fermenting mannitol better than the conventional yeast, as well as fermenting alginate into ethanol. Seaweed as a new source of biofuel, thanks to the reconstruction of E. coli, has become increasingly popular. Synthetic biology really starts to get interesting when we start to understand things such as toggles and biodetectors. A genetic toggle switch is a repression system which can use individual genes to code for the transcriptional repression of other genes. It can be thought of as a light switch. In a room, the light switch allows the light to be on or off. It is either one setting or the other due to an external force. In a cell, a toggle switch can allow the expression of one gene, gene A, or the other gene, gene B, with the use of an external stimulus. The practice of using biodetectors was established in 2007 as a way to detect the presence or absence of biological materials such as pollutants, hazardous agents, and pathogens. An example of the use of biodetectors is the use of bioluminescence in bacteria. Organisms including fireflies, bacteria, marine, and other terrestrial organisms are categorized as bioluminescent species. Bioluminescence is the fluorescent light that is emitted from an organism due to chemical reactions that take place. These organisms create luciferin, a pigmented chemical that when reacted with oxygen emits light. The catalyst luciferase mediates the reaction by speeding it up and is also used in many fields of study such as genetic engineering and detector genes. Researchers are testing different ways to apply bioluminescence, such as detecting bacterial organisms by using certain sensor responses. Despite the many uses of synthetic devices in research, there have been major setbacks in synthetic biology due to their faultiness. Environmental controls necessary to create the desired function of the engineered microbes becomes difficult to pinpoint, and the quality of microbial manufactured parts is questionable. Another issue of bioengineered cells is bistability. Bistability is the variation in the expression or activation of inserted genes. Activation of synthetic gene circuits can depend on the host's physiology. If the correct host is used for the particular circuit, then unintended interactions between the circuit and the host become predictable, and variability can be more easily controlled. In response to the constant issues and failures of these devices, synthetic biologists struggle to document, characterize, and develop quality devices in order to reduce variability of results in different labs. Though these issues are not to be overlooked, with the lowering cost and standardization of synthetic biology, the synthesis of organisms and systems is becoming limitless. Soon, synthetic biology will become a question of what our minds can fathom rather than what we are able to create. As this ethically controversial and complex field advances, so will its definition. As for now, synthetic biology can generally be defined as the act of redesigning organisms or biological systems from scratch to create improved and desired systems that will function in ways unfamiliar to nature.